Yeah, welcome participants. On behalf of Annakkili Amma Research Institute, Chennai and e-teaching Tamil, I am happy to welcome you all to 11th session of aari Bioscience webinar series. I am very much proud and happy to introduce today's special guest, Dr. Neelam Mishra, Assistant Professor, Department of Botany, St. Joseph College, Bengaluru, India. She completed her MSc in Botany with gold medal from Deen Dayal Upadhyaya Gorakhpur University and M.Tech in IIT Kharagpur, and she did her PhD in Texas Tech University, USA. She has received Past and Emma Smith Scholarship 2014 in Graduate School, Texas Tech University, USA. She also received Study Abroad Competitive Scholarship for consecutively 4 years in Texas Tech University, USA. She has research experience of more than 10 years in the field of plant genetic engineering, plant physiology, molecular biology, and medicinal plants. She also contributed to the field of science by publishing her research articles in the reputed international science journals. With this short introduction, I welcome Dr. Neelam Mishra to inspire students with her valuable presentation. Madam, stage is yours.
Good afternoon, everyone. Thank you so much, Mr. Perumal, for your kind introduction. In spite of this unique pandemic situation, it's really great. Oh, is it? Okay, I'll just increase that. So, in spite of this unique pandemic situation, it's really great that we all are connected globally and we are learning and sharing our knowledge, right? So it's a great honor, and I feel really immense pleasure in sharing my research idea on this platform. And I would like to request all the participants to keep themselves mute at all times. And if you have any questions, you can post it in the chat box, and which will be definitely answered at the end of the presentation. So I'll be sharing my screen now. Sir, can you confirm if my screen is shared? Sir, can you confirm please if my screen is shared? Okay, okay, thank you so much. So today I'll be talking about the how to improve the abiotic stress tolerance in plants via genetic engineering. So before I actually start my topic, how to improve the tolerance, I would like to introduce about a little bit about the details of abiotic stress and a little bit of background why we are doing this research. So abiotic stress, as you all know, this accounts for all the non-living factors which are basically affecting negatively the plants, the growth and development of the plants, right? So all the non-living factors basically which are required for the growth and development of the plants are, you can say, light, water, temperature, humidity. So on the basis of what living factor is in excess or is it in suboptimal amounts, that defines that there will be different kinds of abiotic stresses that is faced by the plants. So there are different types on the basis of which non-living factor is being affected or is affecting the plants growth and development. So first of all, there is a low availability of water, or if there is a major excess of water in the field, you will see either drought or flooding stress. If there is too much of salts in the soil, you will see salt stress. Then if there is too much of temperature or too low of a temperature, you will see either heat or chilling stress. Then apart from this, there are several other abiotic stresses which affects the growth and development of plants. First one is radiation stress, then mechanical stress, which is caused by wind, and then there are chemicals. In particular, chemicals can be either macro or micronutrients or heavy metals like copper or cadmium or other kind of toxic heavy metals. So basically in my talk I'll be talking the ones which I have made it bold: the water stress, in particular drought stress, the salt stress, the temperature stress. I'll be talking both excess as well as the low temperature, that is chilling stress. So as we all know that abiotic stresses there in the plants. Plants are sessile organisms. They cannot move away when they are facing any kind of abiotic stresses. So what they need to do is they need to either acclimate or they need to either adapt, right, to make sure that they can survive and they can reproduce and they can complete their life cycle, right, without much yield penalty. So there are definitely so many responses which are displayed by the plants so as to make sure that they can survive and reproduce, though with a little bit of yield penalty. So we all know that abiotic stresses are multidimensional. They can cause the responses at all morphological, physiological, biochemical, as well as the molecular level. Since I'll be talking majorly about drought, heat, salinity, and cold, so I have just taken a picture from one of the papers that was published. So it is basically morphologically you will see the effects in the plants in terms of reduced growth. The roots and shoots in the plants will be decreased, right? The plants will be shorter in size. They will be having a reduced crop yield as a result of that, because of one of the reasons, primary reasons might be early senescence or premature senescence. Now, there are a lot of physiological responses that will be shown by the plants as a result of this abiotic stresses. They will be not be able to uptake water, right? Or there will be whatever water is being uptake, it is not efficiently being utilized to produce the food. So it basically it will be having reduced photochemical efficiency, then altered transpiration, then decreased or reduced nitrogen assimilation, and also metabolic constituents will be accumulated in a high concentration or the metabolic activity will be decreasing. Now, apart from all the physiological and morphological responses, there are some biochemical responses also in the plants as a result of abiotic stresses. The plants will show an increased amount of ABA, which is the one of the major stress hormone that you will see throughout my talk. Then there are certain biochemicals like osmoprotectants like proline, mannitol. They will be increasing in size. Increasing in the amounts, so that the plants can basically tolerate that osmotic stress that is happening inside their cells. Apart from the biochemical effects, there will be so many molecular responses which will be there in the plants, right? Due to drought stress, what will happen is the gene expression will be altered. I mean to say that several genes will be upregulated and downregulated due to drought stress or heat or salt or cold stresses. So that also needs to be looked on. And so many enzymes or proteins will be synthesized, some will be degraded. So all these kind of are the basic responses that you see in the plants due to the abiotic stresses. So as we all know that it's basically abiotic stresses is basically affecting the homeostasis in the plant. So all these responses are basically affecting the plants to perform at an homeostasis level, right? Move on to the next slide. As we all know that pictures speak louder than words, so that's why I put some pictures here. The first picture is, it's taken from one of the land in China. It's basically the excess overuse of fertilizers. They have made these salts to even come out of the soil, and now that soil is no longer good to grow crop plants. The second picture I show you here is taken from somewhere in Maharashtra. Because of the heat stress, what happened is the plants, they have almost died. They are dried, all the chlorophyll has gone, the photosynthesis has gone, and also the yield. Some of the plants are, because of that, some of the plants are dead also. The last one is the drought stress. This picture is taken from the recent drought that happened in Maharashtra several years ago. So what happened is, because of that low availability of water, the soils were completely cracked, and then they were not able to grow plants on that soil for few years also. So a little bit of this abiotic stress, we all know that our agriculture, doesn't matter whether it's India, US or China, it's already almost dependent on the climate. And we all know that we all are facing climate change. So abiotic stresses are basically causing more than 50% of the crop losses due to the climate changes. And we all know that 70% of the water that is available on earth is used basically for agriculture only. And it has been projected that climate change, which is basically what you will have is high temperatures over the years, and also like infrequent rainfall patterns, or maybe no rainfalls at all for several months. So it will decrease the crop yields by 20% or more by the year 2050. It has been already projected. So we all know that plant breeding is already doing its job. They are over the decades, over the last 20 years, 30 years, plant breeders are, trying to develop a variety that can survive abiotic stresses. It can be the drought, salt or heat or whatever. And but it is not sufficient enough to cope up with the crisis. We know that population is increasing at a very high pace. By the year 2050 will be almost increasing at a very faster pace. So we all know that so as to meet the increasing population demands, plant breeding approach is not sufficient enough. We need to think of something that is called as the transgenic or genetic engineering approach, right? So as to make sure that everybody in the world is properly fed and clothed. This is one of the pictures that I want to show you here. Is it was published somewhere in one of the books, that the intensity of the color here shows that what is the chance that the summer average temperature will exceed the highest temperature that is already there on the record, right? So if you see, a red color is basically there is more than 90% chance that summer average temperature will be higher than what usually it is there. And you will see on the globe itself, more than 70% of the area is depicted as red. So there is likely a chance that because of the climate change, the heat, the temperature on the earth will definitely increase. Whatever summers we have now, definitely after 50 years down the line, it will be, the temperatures will be even much higher. Now, what was our approach, right? This was basically the background why we wanted to do this kind of research. So we wanted to improve the abiotic stress tolerances in plants, and our approach was basically the genetic engineering approach. How we used to go about it is, we used to select a gene of interest and then clone it. Either we used to clone it by ourselves, or we used to get the clone from our collaborators, right? So we used to get the E. coli clone. Then after that, we used to transform this the clone into the agrobacterium by freeze thaw method. And then we used to transform the plants with the agrobacterium mediated gene transformation. We use that indirect approach, not the direct one. We use the agrobacterium mediated gene transformation. And then once we get our transgenic plants, we used to analyze it at the molecular level, right? Which means we used to see whether the genes are present in the transgenic plants or not, whether the gene of interest that we try to insert is there in the transgenic plants or not. So we used to do it by PCR. Then the second thing we used to do is, we used to see whether that gene, whether it's there, but whether it's properly expressed at RNA level or not. So for that, we used to do Northern blot. I'll be showing you some of my results. Northern blot and real time, and also sometimes reverse transcriptase PCR. Also, in some cases, if it's an enzyme that we cloned it or any protein that we cloned it, we also used to see whether that is expressed at the protein level or not by using the Western blot analysis. After confirming that our gene is there in our transgenic plants and it is expressing both at the RNA as well as the protein level, we used to see whether it's performing better in the greenhouse and in the controlled chamber conditions. So we used to provide the control conditions, the optimal ones that actually the plant needs for their growth and development. And we used to also provide them with these stress conditions. And that is possible in the greenhouse with all those kind of control things. So we used to do that, and we used to analyze all the physiological parameters like relative water content, or photosynthesis, or transpiration, or stomatal conductance, or several biochemicals. We used to extract the biochemicals, and we used to do the assays, and all those kind of things we used to do. The last thing was, we used to see whether some of the genes which are already been known to involved in the abiotic stress responses, whether they are upregulated in our transgenic plants or not. And by doing so, we were able to kind of provide a model that how our gene will be affecting, or how our gene is able to improve the stress tolerances in plants. And then lastly, which is definitely required for any kind of crop plant, is we used to go for a field trial. And field trial, we used to do it for at least consecutively 2 to 3 years and make sure that whatever our plants are performing in the lab or in the chamber conditions, it is basically, whatever we are performing, it's basically doing the same thing in the real world itself, right? So this was our approach for creating and analyzing transgenic plants. So I was working in Dr. Hong Zhang's lab in Texas Tech University, and our lab was basically focused around creating, doing all these things only in the last 10 years or 6 years of my PhD. Actually, I was doing all these things only. So definitely there are so many genes that that are already available in literature that have been proven to improve the abiotic stress tolerances in plants. But the genes I'll be showing you in my subsequent slides is the one that I have worked on and I have published my research. So those are really promising in terms of greenhouse trials as well as in terms of this field trials. So there are several genes that I have worked on is AVP1, IPT, NHX1, SOS1, CIS1, PP2A, C5, RCA, and AKR2A. So what I will be doing is in my subsequent slides, I'll be talking about the gene, what this gene stands for, what it does, and then what is its mechanism of action, how does it improves the abiotic stress tolerances, and then I'll be sharing with you what we did in our research and how we published our papers. What was our research? And our lab was basically, it was working on the model plant Arabidopsis. We used to introduce all these genes first into Arabidopsis because it is easy to transform and it's easy to see the results. It's very quick to see the results. In a couple of months, you'll be able to get the transgenic plants and also analyze those. And once that proof of concept is done, we used to transform our crop plants. The major two crop plants that I have worked on is peanut and cotton plants. So my next subsequent slides will be about each genes. So first of all, I want to talk about AVP1. This was one of the most promising gene that we have ever seen in improving the drought as well as salt stress tolerances. As we all know that abiotic stresses will never come alone. So we always wanted a gene that can confer the more than multiple stresses, stress tolerances at the same time. So this is one of the genes, one such gene that can confer multiple stress tolerances at a single time. So AVP1. AVP1 is an acronym for Arabidopsis vacuolar pyrophosphatase, right? So this gene is basically, this pump is located on the membrane of the vacuole that is called as tonoplast. And if you can see this picture, this is located on the membrane of the vacuole, and it utilizes the energy of the hydrolysis of the inorganic pyrophosphate. The hydrolysis of the inorganic pyrophosphate to orthophosphate, what will happen is it will create an electrochemical gradient of H plus ions inside the vacuole. So what will happen is, as a result of AVP, right, the concentration of H plus ions in the cytosol will decrease. However, the concentration of H plus ions or protons will increase in the vacuole. So can we say this one is an vacuolar acidification kind of process, right? So this is one of the genes that we have used for, and this proton pump is definitely, it's not energized by ATP. This is energized by inorganic pyrophosphate. That needs to be kept in mind. Now, this gene has been basically proven to be very effective in improving both drought and salt stress tolerance. The next slide will be looking at how it improves the drought and stress tolerance. This is just a pump, right? An AVP gene, it's an endogenous gene, is there in every plant, right? We just try to overexpress this gene. We are taking it from Arabidopsis, putting it into peanut or cotton plants and overexpressing this AVP1. So you will have more and more of H plus ions inside the vacuole. So vacuole will be very much acidic in your transgenic plants as compared to those of your wild type plants which do not have that transgene. So what are the roles of AVP1? How this AVP1 is improving the salt stress tolerance? As I told you, because of the hydrolysis of the inorganic pyrophosphate, the proton is being pumped inside the vacuole. An electrochemical gradient is established for H plus ions. This will basically, what it will do is, it will drive the sequestering of sodium ions, and this is another pump that is situated on the vacuolar membrane. It is called as ATNHX1, that I'll be talking later in my talk. So it's basically, this is an antiporter which transports the H plus ions, and at the same time it sequesters these sodium ions inside the vacuole. We all know that, all the plant cells, inside the plant cells or inside the cellular compartments, that that redistribution of ions or reducing sugars and amino acids, that always takes place, right? So as to make sure that the concentration of the ions and sugars or amino acids or any biomolecules is maintained properly. So here, by doing so, what we are doing is, this AVP1 is directly affecting this sodium proton antiporter. So more and more protons are there inside the vacuole, that will drive this antiporter to import more and more of sodium ions inside the vacuole, right? As we all know that salinity, if we consider sodium chloride, it's the most harmful, salt that might be present in the soil. So sodium ions are really toxic, right? They can disturb the enzymes which are there in the cytosol. So if we can make a transgenic plant that can overexpress AVP1, so that overexpression plant will be able to sequester more and more of sodium ions inside the vacuole, due to which the toxicity of the sodium ions inside the cytosol will decrease. In the cytosol will decrease. However, all the sodium ions will be sequestered, which is the toxic ones, will be sequestered inside the vacuole, right? So as a result of this, what will happen is, no matter you can grow the plants in higher saline soils, what will happen is, these sodium ions, when they come in the cytosol, because of the overexpression of AVP1, it will drive the expression of, it will drive the import of this Na plus ions inside the vacuole. So plants can basically grow better in the saline soils because they can sequester sodium ions better than the wild type plants. So this is the first picture that I shown you, is telling you how these plants are improving the salt stress tolerances. Second picture shows how the AVP1 is involved in improving or regulating the drought stress tolerance. So AVP1 is basically present on the vacuole, and then it somehow it affects the H plus ATPase, which is present on the plasma membrane. So if you have more and more of AVP1, the expression of H plus ATPase, which is another pump that is driven by the hydrolysis of ATP, it will make sure that H plus ions will come out in the apoplast. So apoplastic pH will be decreased. Because of the decrease in the apoplastic pH, the auxins, IAA, it's indole acetic acid, it's one of the auxins, they get protonated, right? And then their movement is basically increased, right, because of their protonation. So these protonated auxins, they will come inside the cytosol, and with the help of PIN1 transporters, they have also shown that this overexpression of AVP, it somehow impacts this PIN1 transporters. So these are nothing, these are the auxin transporters, which basically directs the polar auxin transports and is responsible for increased root growth inside the plants. That's what we need in terms when we have a low water in around the plants. We want the plants to grow longer and profuse root system so that they can search for water deep inside the water table, right? So this is the way that AVP is improving the drought stress tolerances in the plants. Now the second one, AVP is also involved in so many other kind of pathways. So there are several metabolic reactions which are happening inside the plants, is like synthesis of DNA or all those kind of nucleic acids or carbohydrates. They generate inorganic pyrophosphate, right? So all these reactions are generating the inorganic pyrophosphates. They are generating the inorganic pyrophosphates as a byproduct. What AVP1 does is basically it is removing whatever is generated by these processes. This AVP1 is removing that inorganic pyrophosphate and converting it into orthophosphate. So why this AVP1 is helping, or how it is helping, is if it is not utilizing this PPI or inorganic pyrophosphate, this might act as an inhibitor for various metabolic reactions, right? Feedback inhibition, if you remember, right? So inorganic pyrophosphate, if they are in larger amounts, the metabolic reactions will be kind of decreased or reduced. And also it enhances the gluconeogenesis, which is one of the pathways that utilizes PPI. So in some ways, it is basically promoting all the metabolic activities inside the plants. The last one is it mediates the sucrose phloem loading, right, which is definitely required to maintain the source and sink relationships. So as you all can see that AVP1, which is here on the this H plus ATPase, it transfers this H plus ions, and then this H plus ions are basically importing, right, the sucrose with the help of sucrose proton symporters. Right, so sucrose is loaded inside the phloem companion cells, and due to which the source and sink relationships is being maintained, and from the leaves the the metabolites or the sucrose which is being synthesized is transferred to the roots, and that's how that increased root growth is happening. At the same time, because of that acidification of this H plus ATPase, the acidification of the rhizosphere happens, right? Because of the acidification of rhizosphere, the roots, the nutrients which are present in the soil, in the water, they will be properly mobilized, right? So these are various roles of the AVP in drought and salt stress tolerances, or in different kind of abiotic stress tolerances, right? So what we thought is this AVP1, it's been last 20 years, most of the people have worked on first research. It was done on tomato, and it was a very good paper. It was published in PNS. So what we thought of is, we thought of introducing this AVP1 into peanut. And we published this paper in Plant Biotech Reports back in 2013. And what we did is we produced our transgenic peanut plants overexpressing the Arabidopsis gene, vacuolar pyrophosphatase, and we tried to see whether this peanut is having improved, or whether it can outperform in field conditions under drought and salt stress conditions. So I'll show you some of my results. All the results cannot be shared here, but you can always go through my papers and see the results. But some major results I'll be showing you here. What is the performance of transgenic plants under abiotic stress conditions? As we all know that peanut still is number 5 of, ranks number 5 in the major oilseed crops that we have. And then China, India and US, these are the major 3 peanut producing countries, right? But all these 3 countries are facing this issue of drought and salt. Because of the drought and salt, the crop yield is very less. That's what our idea was, to improve the the yield of peanut by introducing this AVP1 gene. So we produced our transgenic plants, and then we tested them under abiotic stress conditions, under normal conditions, which is very important for any kind of transgenic research. Under normal conditions, these plants should not perform different than the wild type plants. Otherwise, you have to take a relative approach. However, under normal conditions, our transgenic plants were performing the same as the wild type plants. But when we expose them to the abiotic stress conditions like drought or salt, what we found is the wild type variety, in which there was no transgene, there was a significant reduction in the shoot weight as well as the root weight, both dry and fresh. Also, we measured some physiological parameters. In the senses, we measured the photosynthesis. We also tried to measure the stomatal conductance. We found that our transgenic plants, the conductance was much higher. They were having higher transpiration, but at the same time, they were having higher photosynthesis as well. And there was a significant increase in the photosynthesis in terms of as compared to that of wild type plants. NT here stands for null transgenic in the senses these plants, they were created from the tissue culture. When until we get the homozygous plants, we screen our transgenic plants. So whatever seeds we get, we always screen, and then whichever is coming out non-transgenic from there is basically referred to as null or non-transgenic. That is what is NT, or in my full talk you'll be seeing this picture, that wild type and SNT, and then these are 1, 2, 4, 6 is basically nothing. These numbers, they represent the independent transgenic lines that is having the AVP gene. Also, as we know that this is an oilseed crop, so we wanted to make sure that our transgenic plants, whether there is an the fatty acid content or the oil content of the plant, whether it is decreased or increased, or whether there is a major change, what we found is there was not significant increase as well as there was not significant decrease also. So the quality and the nutrition of the peanut was exactly the same as the wild type plants. Also, we did our field trial in the low irrigation or just the rain-fed, plots. We did it for consecutively for 2 years, and we found that all the transgenic lines, independent transgenic lines, they always performed better than the wild-type plants in terms of yield of the peanut, right? So, how much grams of peanut we are obtaining per plant, we tabulate that, and then for around in a plot of around we had around say around 250 or 300 plants, and we used to do that, how much grams of peanut we obtained for per plant. So what we found is definitely this AVP gene is very much promising in terms of it improves the, definitely it is not performing the way the peanut should perform in a normal condition. Yield penalty is always there because you are providing stress, you are giving stress to the plant, but the yield penalty is much less in the transgenic plants as compared to those of wild type plants. Having seen that this AVP1 is performing very nicely in the peanut plants, though I was not directly involved in this project, our lab also introduced this AVP1 into the cotton plants, and they have found the similar results in the cotton plant as well. So the first picture is basically they grow the plants under salt stress conditions. You can see wild type plants a little smaller, they are paler also. However, the last 2 rows are basically the transgenic plants that we grew in the greenhouse. They are bigger in terms of their shoot weight, the leaf size, the green part of the leaf is also more, and in terms of root also, they were much bigger under salt stress. In drought stress, what we saw is they are having bigger roots, longer roots as compared to those of wild type and segregating non-transgenic plants, which is referred to as SNT. Though I was not directly the part of this research, but since I was in the lab, so I just want to show this paper also was published in a very reputed journal, Plant Biotech Journal, back in 2011. And they found similar results here also. The shoot weight, fresh as well as the dry one, is increased, and the bole number in the plants, which is shown here, it was significantly high. The numbers show the independent transgenic lines, and the bole number was high, and also the fiber yield was, because cotton is major fiber crop, right? So the fiber yield was also significantly higher in transgenic lines. Now the second gene I want you to take you is the IPT gene. IPT stands for isopentenyl transferase. And this gene that we have used in our research came from Agrobacterium tumefaciens, which is basically a natural genetic engineer. It can basically naturally infect the plants, right? So what this IPT does, this is an enzyme that is basically involved in the cytokinin biosynthesis. It's a rate limiting enzyme in cytokinin biosynthesis. This is the picture I have shown you here. This is the Arabidopsis IPT. This follows this pathway to synthesize the transzeatin, which is one of the cytokinins. And this agrobacterial IPT, however, it takes a shorter pathway and converts the diphosphate to monophosphate, and then consequently that transzeatin is being synthesized. So it's basically an enzyme that is involved in the cytokinin biosynthesis. As we all know that what is the role of cytokinin is, we all know that plants, they undergo that's genetic programming. Whenever they are under stress, that genetic programming of senescence and death is being accelerated, right? So either you can have two different kind of approaches. You can either overcome or suppress that senescence that is happening, which can be done with the help of this cytokinin. Cytokinin is a hormone. It basically delays the senescence, premature senescence it will delay. Or other approaches, basically you can study all the epigenetic mechanisms which are there, and you can study all the genes which are involved in the early transition from vegetative to the reproductive stage, and basically use that knowledge to avert or to basically divert the plants from early transition to flowering, right? So what we did is this IPT plants, what is the mechanism of action? I'll just brief you here how it works in the plants. So as we all know that cytokinin, under normal conditions, what will happen is the balance of cytokinin is maintained because of these two enzymes. IPT is involved in the synthesis of cytokinin, biosynthesis of cytokinin. However, CKX, which stands for cytokinin oxidase, it is responsible for the degradation of a cytokinin. In a normal plant, the balance is being maintained. However, under optimal conditions. However, under abiotic stress conditions, what will happen is the CKX under osmotic stress or any kind of abiotic stresses, this expression or the enzyme activity of this CKX will increase, right? So what will happen is cytokinin will be basically the high amounts of cytokinin will not be there, and there will be decreased amount of cytokinin. Normally cytokinin, what it does is basically it's a antagonistic to ABA or other kind of hormones. It basically what it does it induces if there is high amounts of cytokinin, if there is you know high amounts of cytokinin because of less amounts of CKX, what will happen is high amounts of cytokinin will repress the ABA, ethylene or reactive oxygen species production. That's what we want, right? So what we'll do is we will overexpress IPT so as to counterbalance the effects of CKX, right? CKX is inducing ethylene, ABA and ROS. What we'll do is we'll try to repress the synthesis of ABA, ethylene and ROS. In doing so, what we'll do is if we are repressing the synthesis or biosynthesis of ethylene, the nutrient will be mobilized at a greater rate, right? Improvement of source and sink relationships will happen, and then the senescence will be delayed. That's how we can basically delay the senescence, and we can make sure that plant can complete its life cycle, and even though yield penalty is there, we'll get something out of it and we'll get some crop yield, even though at reduced levels, but at least something is better than nothing. Then the other thing is it will reduce the amounts of ABA. So all the ABA induced events, which is basically the ABA is there, it will decrease the the opening of these stomata. It will cause these stomata to close. It will decrease the photosynthesis. So if you increase cytokinin, all the ABA induced events will be suppressed. So you will somehow be able to maintain for a period of time, you will be able to maintain the photosynthetic efficiency of the plants. Also, it will repress the reactive oxygen species. How it will repress the reactive oxygen species is it will basically improve the activity of several antioxidant enzymes like peroxidase or superoxidase or catalase, all those kind of superoxide dismutase, all these kind of enzymes, the improved activity will happen, right? So this IPT is making sure that cytokinin levels are high, but at the same time we want to make sure that since it's a hormone, this level should not be high at all times. It should be regulated. So we wanted to express this gene but in a regulated manner. So that's why what we did is we express this gene under the control of a water deficit and senescence associated promoter.
Audience, kindly wait 2 minutes. Madam will join back.
Can you hear me now, sir? Okay. 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 Host has disabled participate screen sharing. Can you dis? Sure.